All right, in this video, we're going to talk a little bit about how gene regulation occurs in eukaryotes. So there are a couple questions that make eukaryotes gene regulation different. Number one, how are they turned on and off? Now that's kind of the same question as the prokaryote, but the answer is different. And how do cells with the same gene differentiate to perform completely different specialized functions? How does a skin cell become a skin cell and a muscle cell become a muscle cell and so on and so on? And that's a very different question than we had to deal with prokaryotes, because prokaryotes are all unicellular, whereas many eukaryotes are multicellular, and the cells are specialized. Even though they have a full set of genes, only some of the genes are ever going to be active. So prokaryotes, single-celled, evolved to grow and divide rapidly. They must respond quickly to changes in their external environment to exploit transient resources when they're available. So gene regulation in prokaryotes turns genes on and off rapidly. It has some flexibility. It's reversible. It's used to adjust the levels of enzymes for synthesis and for digestion. Eukaryotes, many of them are multicellular, like we said. They've evolved to maintain a constant internal condition while facing changing external conditions. We call that homeostasis. Pretty much every system in the body of a living thing other than reproduction is for homeostasis. Growth and development, again, is a big deal, where it's not so much so in the prokaryotes. It's a long-term process. It involves specialization of cells, turning on and off large numbers of genes in a cell. And it must coordinate the body as a whole rather than serving the needs of individual cells. So where does this occur? In prokaryotes, remember, it was the codon, and you had two kinds. You had the inducible and the repressible codons, and we talked about in the last video how that can work in the prokaryotes, and it does work well for them. But in the eukaryotes, we have various points along the process of going from a gene to a protein where control can happen. And you can see those 7 there, and we're going to talk about each of those as we go through the video. So the first level is the DNA packing level. Remember, in prokaryotes, you got a single circular chromosome. There's no nucleus. It's just in that nucleoid region of the cell. In eukaryotes, you've got multiple linear chromosomes that have to fit in the nucleus of the cell. So how do you get them, all that DNA, all that genetic information in the cell? Well, you got to fold it up, you got to coil it, you got to organize it. So there are different levels of structure in the DNA. You have the basic level, the DNA double helix. You have the nucleosomes. You have the chromatin fibers, the loop domains, and then the chromosomes that we see during cell division. So the nucleosomes are made up of a core protein, or core of proteins, called histone proteins. There are 8 of them that make this spool that the DNA can wrap around. And it looks kind of like beads on a string when we look at it. It's the first level of DNA packaging. Again, the histone proteins are positively charged. DNA is negatively charged, so the DNA is attracted to the histone proteins. And basically, what this does is any genes that are in that coil around the histone are essentially turned off, at least temporarily, because the enzymes and mechanisms for transcription can't get to those genes. So the degree of packing of DNA regulates transcription. Tightly wrapped around the histone, no transcription because, like I said, the enzymes and things can't get in. The genes are turned off. We call that stuff heterochromatin. It's darker when it's stained, it's tightly packed. Euchromatin, on the other hand, is loosely packed. It's between the nucleosomes, and it's lighter when we stain it. Another level in which DNA regulation occurs is methylation of DNA. And we talked about adding the methyl cap, but this is different. Methylating DNA bases, in particular cytosine bases, can block transcription factors. So without those transcription factors to organize and to help the RNA polymerase bind, there's no transcription. The gene is turned off. This particular form of control is pretty much a permanent way. So this would be turning off genes that cell is probably never going to need. If you're a skin cell, you're not going to need the genes for hemoglobin, for instance. Another place we see this is in mammals like us. Females have 2 X chromosomes, but having a double dose of those genes is potentially bad. So one of the X chromosomes gets shut off. It's condensed into bar bodies, and methylation plays a role in that. Another area where we can regulate gene expression is at the histone level. By adding an acetyl group to the histones, it helps to unwind the DNA and make it available to the transcription mechanisms. So this loosely wrapped DNA can be transcribed and then translated later on. So this turns genes on. So this changes the shape of the histones and makes the DNA not as tightly coiled around it as it normally is. Transcription initiation is another level of control. So basically, what has to happen in order for transcription to occur? Well, you have to have the RNA polymerase bind to the promoter. But to do that efficiently, you need transcription factors to position the RNA polymerase, and then you also have the activity of the activators, enhancers, and things like that. So where the RNA polymerase binds is called the promoter, and it's basically a control sequence to the gene. There's also a binding site for the transcription factors that help position the RNA polymerase and connect it with the promoter to allow transcription to occur. So this setup gives us a base rate, a low rate, if you will, of transcription. If we want to turn transcription up to high, that's where the enhancer sequences come in. The enhancer sequences are where other molecules called activators can bind, and they're further upstream from the gene than the promoter. So when they bind the activator, they work with the transcription factors to turn the gene transcription rate up to high. So enhancer sequences, like I said, distant control sequences. Activator proteins bind to the enhancers to stimulate transcription, to raise the rate of transcription. Silencer proteins can also bind to the enhancer sequences, but in this case, they would block transcription. So this is the transcription complex for eukaryotes. You've got your RNA polymerase, you've got your transcription factors bound to the regulatory sequence like the TA-TA box. You've got, in this picture, you've got 3 different activators bound to enhancer regions. You also have a co-activator connecting the activator to the transcription factors, and all of this works together to allow transcription to occur. Third level is post-transcriptional control. So transcription, we've made our primary transcript. Now what can we do with it? One thing we can do is alternative RNA splicing. Remember, eukaryotic genes have introns and exons. So by changing which exons get put into the mature messenger RNA, we can take the same piece of DNA and use it to make different related proteins. This allows the DNA to be more efficient. When we first started looking at the Human Genome Project, for instance, there was 100,000 proteins and the human body makes approximately. We thought there'd be about 100,000 genes. Turns out there's only about 20,000 genes. Because of alternative splicing, we can use the same gene for multiple proteins. Once the messenger RNA is made and we've done all the things we need to get it out of the cell, we've added the 5' cap, the poly A tail, we've connected the exons together. Now it's headed out of the nucleus into the cytoplasm and to the ribosome. The longer the messenger RNA hangs out in the cell, the more protein the cell is going to make. So we don't want to make too much protein, but we're going to make enough of it that the cell can do its job. So messenger RNAs can last anywhere from just a few hours to a few weeks, depending on how much protection we add to the messenger RNA before it leaves the nucleus. And then there's this RNA interference. So basically, there are these what we call small interfering RNAs, which are made by an enzyme called dicer, which takes RNA and cuts it into little pieces. Those little pieces can bind to the messenger RNA, which creates a double-stranded messenger RNA. Double-stranded messenger RNA can be tagged or marked for degradation. Also, these small interfering RNAs can cause gene silencing, which is a post-transcriptional control where the genes are turned off and no protein is produced. So here you can kind of see the process. Dicer producing the small interfering RNAs, which bind to the messenger RNA to make double-stranded. RISC is the enzyme that breaks down the double-stranded RNAs and shuts the gene expression off. So this was discovered by two gentlemen, Andrew Fire, Craig Mello, in the 1990s. They got a Nobel Prize in 2006 for their discovery of RNA interference. So fifth level of control is called the control of translation. So we've done transcription, we've modified it, we've sent the messenger RNA out of the nucleus, it's got to the ribosome. So one of the things that can happen is we can block translation by blocking the initiation process. So there's a way to prevent the initiator tRNA from binding with the ribosomal subunits, which then blocks translation of that piece of messenger RNA. Step 6 and 7, protein processing and degradation. So once the protein's made, things have to happen to the protein. That's the processing. And the proteins, like the messenger RNA, aren't going to hang out forever, so they are eventually broken down when the cell no longer needs them. So protein processing, once the protein is produced, it has to fold, it may have to be cut, we have to add things to it like sugar groups, we have to get it from the ribosome to where it needs to go to do its job. Maybe that's somewhere in the cell, maybe that's exporting from the cell to do its job somewhere else. Protein degradation, proteins that have done their thing and we don't need anymore, they get marked by a molecule called ubiquitin. Ubiquitin is a marker for a structure called a proteasome, which is basically a protein structure that acts as an enzyme. And when it finds a ubiquitin marked protein, it will basically pull it in and cut it up. And those amino acids and those polypeptides or those peptides can be reused to make other proteins. So this is called proteasome degradation. 3 different researchers, 2 in Israel, 1 at University of California, Riverside, worked on this ubiquitin thing, and they basically figured out how it worked in the 1980s. Again, Nobel Prize in 2004. And that kind of finishes up our discussion of eukaryotic gene expression control.